Um, Thank you so much. I deeply thank the organizers for it's, it's, deeply thank the organizers for inviting me to this one-day meeting. It's a privilege on many levels for me. On one hand, of course, Sri Shankara Cancer Hospital has become a second academic home for me in Bangalore after the Indian Institute of Science, because so much of what we do starts here now, goes through processes that are done here, and intellectually is strengthened by the kind of science that we attempt to do in my group and laboratory. The second privilege, of course, is the fact that I'm following up after Shiva, my fellow medical college alumna, and the next time this happens, I'm going to request a break between my talk and hers, because there's just so much that I'm trying to digest from this spectacular presentation that is here. I'm trying to adrenalize myself at the same time, trying to deal with the deep, overwhelming feel that I have for what I heard. The third privilege, of course, again, to be part of a meeting, even a one-day meeting, where we have talks by Shiva, by Sharmila, and later on in online by Pritha. Because these people, when I joined the academic milieu of India, and was wanting to work in breast cancer and set up a program in breast cancer, starting up ovarian cancer was challenging, because I'd never dealt with the literature at that time, and in that sense, reading up the papers, reading the efforts of this very small community of researchers in ovarian cancer. This community was instrumental in providing me with tools, thoughts, support, guidance, and mentorship. And so to be, again, part of a meeting, to be able to talk about some of the work that we do in this gathering, Pritha might be here, I wish she was here in person as well, is again deeply humbling and a privilege. So with that, I'll try to talk about some of the things that we have done, much of which will be unpublished and a little bit published along the way. Optimize? Excellent, thank you. So as I said, much of this is work in progress. So it's to be taken with that, and the critique and the advice and suggestions that we deeply welcome. So we as I look upon myself, I was trained, of course, after my medicine in developmental biology and morphogenesis, and I approach science even when I deal with problems of cancer from the background of developmental biology. So therefore, it stands to reason that when I look at the transformation of organs or tissues from their homeostatic state to a malignant transformation, I look at it as a problem of pattern formation. And therefore, if we our development biology is, of course, involved in trying to understand how organs form and what are the rules of patterning that lead to cells arranging themselves to give rise to the stereotypical patterns that you see, which distinguishes a liver histology from perhaps a pancreatic histology, to give an example. But then these same rules are therefore subverted, they're abused, and that's what leads to the dissolution of the organ and tissue architecture when you have transformation and you have carcinogenesis. But one can, again, as a pattern theorist, turn that problem on its head and ask, if therefore you have a dissolution of that organ architecture, what was referred to as behavior by Dr. Srinath, and again the tumor stroma interaction by Asima, again to link these ideas, can one therefore imagine what one gets in carcinogenesis and carcinomatosis, the dissemination of cancer, as a rejuvenation of new roots, of new form, of new morphologies, and it therefore harks back to that word neoplasm, which in, of course, in Latin means new mold, and therefore we are trying to therefore ask, does carcinogenesis and carcinomatosis therefore represent a new set of rules that the tumor now uses to interact with its stroma, that the cancer cells now use to interact with the extracellular matrix, that the cancer cells use to interact with immune cells, stromal cells, fibroblasts, and the rest goes like. And then, therefore, if we are able to identify these new rules for neoplastic behavior, can we therefore use this to try and move towards being able to devise new therapeutic efforts and attack those specific rules to therefore devise, perhaps in the future, new therapies for cancer. Therefore, with this intent in mind, we have, using both breast and ovarian cancer as examples, been looking at the different locales where cancer tends to therefore behave in different ways from normal, developing, or homeostatic tissues. And of course, be the early migration from the primary focus, the introversation into vascular environments, into circulatory environments, which include not just the blood, the lymph, but also circulatory environments like the peritoneum, the pleura, and so on. The survival of these cells within these environments, where the cancer cells are now exposed to new stresses, new biophysical and biochemical cues that they have to survive in order to, in the end, decide whether they would want to settle down in a new home. How would they now compete with cells in that new environment, and how would they now start thriving in that environment, the process of colonization and metastasis. So we've done a lot of fundamental work in trying to identify some of this new grammar of the new form, some of the new behaviors, and some of the altered behaviors that cancer cells evolve in order to adapt, in order to tide over these new stresses, in order to interact with their new partners, and then therefore survive within this area. But what I will be talking about today is, given the mandate of the meeting, some of those efforts to sort of take that just a tiny step beyond this grammar, tiny step beyond the new histopathological understanding that we have gained from cancer. And therefore, what I would like to harp on is some of the work that we did in the fundamentals of our understanding of ovarian cancer spheroids. Now, this is the sort of the transitory niche, the circulating cells that we talk about, which would otherwise move through in case of breast, prostate, pancreatic cancers within the blood and the lymphatic vessels, but in case of ovarian, in case of ovarian cancer, is found in the peritoneal fluid, leading to the buildup of what is known as malignant ascites, a fluid that is rich in a very interesting biochemical, biophysical way, but also consists of disseminated clusters of ovarian cancer cells, of course, starting from the fallopian endometrium, but entering into the fluid peritoneal space. And these cells tend to cluster together and have been well documented to be able to give rise to metastasis resistance in case of ovarian cancer, especially the high-grade CS ovarian cancer. These were some of the, this is the question with which we started fortuitously. When at some point of time, and this was the time when I was working with clinician collaborators in Kidwai, and subsequently has been validated in Shankara as well, where when we received these circulating tumor cells from the hospital, and we were just putting them under the microscope, it struck me with wonder when I started seeing that these spheroids seem not just to be simple aggregates of cells, but seem to have a level of organization that was much more than just a cluster of attached cancer cells. And I've seen this repeatedly under my microscope several times, that they seem to have this very interesting hollow structure. Now, to a developmental biologist, a hollow structure implies organization, a hollow structure implies patterning, a hollow structure implies, therefore, new form. And the reason why that is so is because hollow structures, as would be known to all of you who look at any kind of glandular histology, implies the presence of cell polarity, implies the presence of tissue polarity. And what is polarity doing in cancer? Where we typically again understand cancer as a dissolution of the organ architecture, which implies the dissolution of the polarity, and hence the grading and all of the literature associated with grading. So, more work subsequently, of course, over the next few years, made us understand that ovarian cancer spheroids from patients and from other models of both in mice as well as through cell lines, seem to transit between these two morphologies of sometimes having this grape-like structure, what we subsequently call moruloids, in deference again to developmental biology, where you have an embryonic form called the morula, which is mulberry, a grape mulberry existence, and subsequently the hollow structures which we call blastuloid or blastula-like. And we could see that the transitions were occurring in a very dynamical fashion between these two morphological states. This is a scanning electron microscopic image of the moruloid, and you can appreciate the cleavage furrows that you see between the cells as they form this mulberry-like structure. And here is a scanning electron microscopic image of a spheroid which has a blastuloid morphology. We found them in patient spheroids, we found them in cell-like spheroids, we found them in murine spheroids, and now, of course, it is, from what I know, people tell me that, oh, look, we see this in pancreatic spheroids, pancreatic cancer spheroids with metastasized peritoneum, we see that in colorectal spheroids, and we've seen them in colorectal cancer spheroids as well. So this is not something that is very specific to ovarian cancer spheroids. We are now realizing aspects of what we have discovered as a generic morphological signature in many spheroids. That's fine. So in the blastoid spheroid, what you can see is, of course, with the scanning electron, which looks at the surface, you can't see the hollow inside, which we have been able to use confocal microscopy to look at. But what comes out here is this sense of a coat that seems to coat these spheroids, and you can see that coat that is there on the surface. Asima again mentioned matrisome, and that's what's coming up. So we did, again, and this is now then subsequently in collaboration with Annapurna and a team at Shankara, we started dissecting these spheroids at a molecular level and using proteomics, using quantitative proteomics, as well as using classical immunohistochemistry, we were able to identify matrisomal signatures associated with blastoid spheroid. That coat that we saw was none other than basement membrane matrix. Again, basement membrane matrix, as per our understanding of cancer, is what is destroyed as the first step for dissemination of cancer in glandular cancers, breast cancer, pancreatic cancer, prostate cancer. Here, in transit, it is brought back, new form, new granule. Okay, so laminin staining, collagen 4 staining, the typical basement membrane markers seem to be present on the surfaces of the blastoids. But are blastoids the only one that use matrisomal signatures? What about the moruloids, the ones that are the mere aggregates of cells? No, again, there was complexity there. When we did the quantitative proteomics, we were able to identify fibronectin as a key extracellular matrix player in that mulberry structure as well. So these were not just cellular aggregates, there was rich extracellular matrix within them as well. So there was not just were cells transforming from a moruloid to a blastoid state, not just was the entire structure transforming, it was transforming at the level of cells in terms of polarity. It was also transforming in the nature of the extracellular matrix that they were bringing on or switching off. Fibronectin was up in moruloids, goes down in blastoids. Basement membrane is down in moruloids, goes up in blastoids. So there's a switch happening on multiple levels in terms of molecular expression as spheroidal transformation takes place. Fine, great. We observe this. What's the point? What's the function? What do they do in these cases? So again, using a slate of cell biological studies, we were able to fluorescently label cells in different ways. And what we were able to identify is that the moruloids, the ones which are grape-like, have a morphological lability. They are able to accept cells within them throughout cells and are constantly in a state of morphological flux. On the other hand, the blastoid spheroids were almost like organs. They would not accept cells which will be present around them. They would not be extruding cells out, and that is due to this shell of matrix that was protecting them, keeping them in this glandular state and preventing them from accepting this. They almost had transformed themselves into very stable, morphologically stable entities. Were they really stable in some sense? So here's and was the the matrix actually imparting stability to them? So this came through time-lapse videographic experiments we ran, where we took, and this was true for again for all the different types of steroids, look at patient steroids from Shankara, from earlier from Kidwa, as well as cell line models, where we removed that matrix, and when we removed the matrix, the spheroids would go from the blastoid to a mononuclear state. So we remove that shell and it goes back. We then restore it by washing away the enzyme that destroys the shell, and it comes back into a blastoid state. And this is dynamic. This told us two things. One, that the matrix therefore is instrumental to the formation of the blastoid state or the transition between the moraloids to blastoids. It also told us another thing. The hollow that forms is not a necrotic core, which is typically the first default hypothesis for experimental biologists like me to think about any hollow. This is a grammatical hollow. This is an organizational hollow. It comes through cellular reorganization, and that is involved, and the matrix is involved in reorganizing this matrix, in reorganizing the cells back to the blastoid state. All right. So, what is the benefit of having intra-patient interspheroidal heterogeneity? What's the point of having this heterogeneity? Because we would see that in patients from whom a scientist would take out and look under the microscope, we would mostly see the hollow spheroids, but we would also see a mixture of, very frequently, a mixture of the moraloid and the blastoid state coexisting within the patients. Now, the first thing we saw is that there seems to be a benefit for moraloids because they are able to adhere really fast. So these are fast adherent spheroids, and the way we were able to do this is by taking mouse peritoneum and stringing it and creating a pseudoperitoneum-like microscopically imageable platform on which we had human cancer spheroids, and we were able to see the adhesion of these human cancer spheroids on the mouse peritoneum, and the moraloids would be able to latch on much faster. By then, do we need the blastoids? So the blastoids have a different property, and that came through our interrogations at the level of mechanobiology and biophysics, where in collaboration with Professor Prashinjit Sen at the Department of Nanosciences, we were able to look at the elastic properties of these structures, and were able to show that blastoids almost were like elastic balls. They were not only morphologically stable, they were also elastic in nature, whereas moraloids were more like putty, like a atta. So they would deform and they would not regain their shape back. But blastoids would be able to regain their shape very nicely. And this was great. Microfluidic experiments which we were able to run. Microfluidic experiments which we were able to run with both moraloids and blastoids, and you could see that the blastoids are able to move through confined environments in a way such that they would not deform, unlike the moraloids, which you just saw coming out and deforming. The blastoids would regain their shape. It's able to regain its shape. But it was able to regain its shape, and it also showed very low potential for disintegration. So what you have here is a static morphological lability now going into dynamic morphological lability, where moraloids would also tend to disintegrate faster but also adhere, whereas blastoids would tend to remain in the peritoneum for longer periods of time because they have poor adherability but they have greater resilience. And we feel that is a very important trade-off that these alternate morphologies are able to achieve. You gain resilience, you lose adherability, you stay in the peritoneum for a longer time, but early metastasis can be mediated by these moraloids, which tend to stay less in the peritoneum, tend to adhere fast, which is why perhaps when we were able to harvest the cytes, you would hardly see the moraloids. Mostly you would tend to see the blastoids. So that's the story of it, and we were then able to, of course, go into cell biological, derive a cell biological understanding of why blastoids have that particular architecture, which is a story for another day. It's an entire cell biological and molecular biological investigation that we have been running. But again, coming to the translational part, or at least my pretentious translation part. What could be, okay, now that we understand this, can we use this in some way to derive a way of, say, breaking the spheroid? So what we did was we used the proteomics, and in this case we collaborated with a mathematician at Translational Health and Science Technology Institute in Faridabad, Professor Samrat Chatterjee, and we were able to derive what is known as a genome-scale metabolic model. Now what this does is this basically takes all the metabolic reactions that can occur within a system and clothes the constraints of our proteomics, tells us that this is the proteomic state that we have in these cells, and maps the possible reactions that must be taking place within individual phenotypes, and that those phenotypes would be monolayers, monocytes, blastocytes. And in doing so, what it does then is it asks, if I were to now perturb certain reactions, would these reactions be enough to break the transition that must be occurring within these states? In other words, getting a sense of whether the dissemination can be blocked by blocking certain reactions. And in doing so, we were able to identify certain key reactions and then look at whether those reactions are druggable. Can we predict drugs that can hit those reactions and therefore block those transitions? And one of the things that we saw was that osentamide, which is a neuraminidase inhibitor, came up as one of the druggable targets because neuraminidase seemed to be involved in being able to move the processes in turn. And this is not super surprising for us because we know that sialic acid and sialic acid biosynthesis, sialic acid biology is crucial to both carcinogenesis as well as morphology. So we were, and therefore, able to look back from experiments into theory, back into experiments, by now adding osentamide to these spheroids before they formed, after they formed, and we're able to break the spheroids after they formed, or able to prevent the spheroid formation before they formed. So this is one way by which we were able to identify a particular inhibitor going from experiments into theory and back into experiments. And we are now trying to test this out by speaking with clinicians to see whether we can start using this. Osentamide, of course, is used in patients for flu, but if perhaps we could take this forward in some way and see if it has benefits in patients who have already a dissemination and are at an advanced stage. The second approach, I have 2 minutes now, I'll weather on faster. The second approach was a larger approach taken using our spheroid assay, because now we are able to look at multiple spheroids, use multiple drugs, and create medium to high throughput screens. And I have a small lab. So medium to high throughput screens where we can look at different drugs and their ability to break spheroids, as well as look at drugs, as well as look at the cytotoxicity of these drugs on the cancer cells. We've developed a screen which we got patented for early this year, and we've been able to identify several drugs that are now we are moving to try and test it again in collaboration, where we can see whether they are able to hit the spheroids, break them, and kill the cells in conjunction and in synergy with existing chemotherapeutic drugs. Now, of course, the more important question is that fine, I can kill a cancer culture. Can I go back? Is it fine? Hello? Is it audible? Hello? I can shout. I think they can hear me. This has a mic, right? The computer should have a mic so they can hear me. I'm sure they can hear me much better than the people. this, again, we've been able to identify a bunch of targets, all of which are repurposable drugs that are able to again prevent spheroid formation if before the spheroids are formed, or able to break the spheroids after they are formed, and of course are able to very efficiently do this in conjunction with carboplatin. But more importantly, again, this is where we wanted to really be careful, because as I was going to say, one can kill a cell culture with a gun and a bullet as well. The point is, would it hurt the cells that are surrounding the stroma that needs to survive even if the cancer is killed? And in order to do that, what we have been trying to do is, yes, that are these drugs viable in terms of their cytotoxicity being specific to cancer cells? And in order to do that, what we do is we again take messenteric cells from individuals. Unfortunately, it's hard to get it from the same patient because the mesentery is often compromised in case of the cancer patient. But we get it from individuals, and we try to see that these drugs, which we see are cytotoxic, are also cytotoxic at the same concentrations on the mesenteric mesothelia, as well as in cell lines of these mesothelial lines. And we see that many of the promising targets are ones which are cytotoxic for the cancer cells from patients, not so for their mesenteric mesothelium. So these are attempts that we are doing, and in the future, what we are trying to do is develop even more engineered platforms where we can interrogate processes that are part of metastasis. This includes intravasation, for example, where we have developed this very interesting intravasation device where we can put cancer cells, endothelial cells, extracellular matrix, almost rebuild the anatomy. My PhD was in anatomy, so that's something I cannot depart from. So we are trying to almost rebuild the anatomy and then trying to study cancer cell, cancer immune interactions within this space. So we've been able to, this is a big paper that is under review and revision in small, as well as patented chip, organ on chip, where we have been able to put in tumor cells, extracellular matrix, endothelia. On the right side, what we are doing is we are trying to move into dynamical organ on chip. So one of the things that we've seen is that the distension of the abdomen, as that occurs with the accumulation of fasciitis, plays also a deep role in altering the cell biology that exists in terms of deciding the mesothelial cancer interactions. This is something that has been seen by Professor Sharon Stack, not a known investigator in ovarian cancer biology, and we are developing these dynamical, distendable organ on chips, peritoneum on chips, in order to study the organ interactions. Where this needs to go into is, and I'm going to take home messages, a combination of models with variable histological and histopathological complexities can, and I hope that, provide deep insight into fundamental mechanisms of how cancer progresses within our real anatomies. They can be used to screen for potential management strategies and investigating the matrix and extendable. I say extendable because we already talked about the cancer immunological aspects that Ashima built on. Microenvironment holds deep potential for moving towards novel target discovery. I will end here, 19 seconds late. I'll end here by acknowledging my group, several of whom are here and are brave enough to be able to embrace both the biophysics that I try to bring in my lab, or the morphological biophysics that I try to bring in my lab, as well as its important clinical implications that will be dealt with whole of today. I would also like to thank my collaborators, Deepa, Roshanjit, Mohit, Barish, and Varun, again, engineers, physicists, biologists, pharmacologists, and of course, none of this happens without the extremely crucial intellectual support from clinicians out here. Dr. Rekha is a long-standing collaborator with my group, Dr. Aruna, with whom we are now embarking on some super exciting discoveries, hopefully in the coming days, if we get funded. And so, as well as past funders who have, of course, funded some of the crazy ideas that we have mushroomed in the lab. And with that, I will be very, very happy to take questions and suggestions. Thank you so much.
As always, a fantastic talk. And congratulations on the progress you made. I had just one question. When you're screening for drugs, do you see a transition between the blastoid and the molecular
Oh,
in
yeah.
response to different drugs?
Yes. So there are two classes, and you're absolutely right. We have identified two classes. There's one class that that breaks transitions, that prevents this transitory nature. The transition would happen almost by default. There are some drugs which revert the transition, and then there are some drugs which will blast the blastoid, which is of course what we would love, because eventually transitions are good for academic questions, but you don't want patients to have these transitions happening. You would want to blast the blastoids. So and one does not need very high concentration. One just needs to understand the cell biology. So for to whet your appetite, we know, for example, that some of the players, the inhibitors of say the Rho kinases, are able to break the spheroids. There are some others, like I think the ARP2/3 inhibitors break, the Rho kinases block. So by tweaking specific and now we have gotten to the cytoskeletal part of how the matrix regulates the actin cytoskeleton inside the cells. By tweaking specific parts of the polarity signaling structure, superstructure, we are able to achieve specific phenotypic outcomes.
Because if unless the transition is broken, I don't think there can be a good outcome.
Absolutely.
Right?
Absolutely.
So I think that is fantastic.
Absolutely. And the neuraminidase inhibitor also blocks the transition, so that is again promising.
I have a question.
Yes.
It's a very interesting work actually. An excellent talk. So, moruloid, then blastoid. How do you connect this to the real carcinogenesis later? Because always we have seen that the cancer will have a necrotic center. So from blastoid, then what happens? Because I always believe cancer is actually a cell's journey back to time travel to early stages of development. That's when cells were without much constraints. So from blastoid, what do you think?
Sure. My humble argument therefore is that it's not always the necrotic core, and it can have a core that need not be necrotic. It can also be an organizational core due to cellular rearrangement. But that's another point.
I
Yes.
agree with you.
Yeah.
I think maybe there is more to it.
The other point that you make, what after the blastoid, eventually what we see is that the myeloids and the blastoids go on to adhere onto the omentum and the mesenteries. We've identified how that happens in mice, which we can image, of course, very clearly, and they are able to settle onto these structures and then spread. It's a different question as to whether the entire molecular signatures are recapitulated from the fallopian era onto the mesenteric era. May not be, and there are some transcriptomic indications that it's not so. But because of the travel that they have done in the form of these spheroids, have given them new memories that they are carrying along at the epigenetic level. So there are interesting patterns. We are in the process of dissecting, but ultimately that is the process that happens. However, the other sort of humble argument that I make is that it's the blastoid structure that contributes to their resilience within the peritoneum, which is subject to a lot of mechanical stresses, including but not limited to the peristalsis that occurs, the convective flows of the peritoneal fluid that occurs, and so on. So that resilience and that inhabitability that these spheroids have, such that whenever we harvest the cytus, we see chock full of spheroids. Why? Why have they not already latched on to the surfaces that are there? That's because of these hollow structures, which have far more to them than just the hollow.
So just like the original blastula, like
Yes.
it has that zona pellucida
Yes.
it should hatch out of
Yes.
and then
Yes.
implant
You
it.
can see the balance.
Yeah.
Yes.
Yeah, it's exactly the same.
So I did not have the SEM image of a blastoid that has settled down, and we see that the shell is cracking.
Will it hatch out of it?
Yes, the shell cracks. I wish I could show you that image. I don't know, it was not there in this particular presentation, but we can see a shell cracking number one. Number two, our transcriptomics indicates that ZP1 and ZP2, the two zona pellucida proteins, are also
they?
being upregulated in stamens. It's crazy, but it's true.
Thank you.
Thank you so much. And the normal cells, yes, there are some very strong signatures. The matrices that we have talked about are mostly present in both, but the levels to which they are expressed are in this case sort of say for pipettin much higher in case of cancer cells. There are certain extracellular matrix proteins that I think are very specific to the cancer structures. I'm just thinking of a bunch of them that we did look at the transcriptomic level, but the names fail me. But I can tell you about those certain systems.